Cellular respiration can be described as a series of metabolic processes that take place within a cell in which biochemical energy is gathered from organic substances, for example glucose, and stored as energy carriers or ATP for use by the activities of the cell. There are four steps for cellular respiration. The first step being glycolysis, the second step being pyruvate oxidation, the third step is called the Krebs cycle or the citric acid cycle, and the fourth and final step is called oxidative phosphorylation, also known as the electron transport chain. Glycolysis is the energy investment phase. It is the process of cellular respiration by which carbohydrates and sugars, specifically glucose, are broken down, producing ATP and pyruvic acid. Pyruvic acid is made by glycolysis in the cytoplasm. However, pyruvate oxidation occurs in the mitochondrial matrix. That means, before chemical reactions can take place, pyruvate must first enter the mitochondria. When inside, three steps are followed for pyruvate oxidation to occur. Step 1. A carboxyl group is nipped off of pyruvate and released as molecule of CO2. What's left are two molecules of carbon. Second step. The two carbon molecules made in step 1 is oxidized. The electrons in the oxidation are picked up by NAD+ to form NADH. Step 3. The oxidized 2-carbon molecule, an acetyl group, is attached to a coenzyme CoA or CoA to form acetyl-CoA. Acetyl-CoA is referred to as a carrier molecule. Its job is to carry the acetyl group to the Krebs cycle.
Right then, the citric acid cycle or the Krebs cycle is one of the most important steps in cellular respiration. I don't think you understand how crucial this step is, guys. Imagine this, if the entire cellular respiration is a Bio20 group, this cycle would be called Earlman. That's how important and vital this cycle is. The Krebs cycle produces the molecules NADH, FADH2, and ATP. These products are produced in 8 steps. Step 1. Acetyl-CoA joins with a 4-carbon molecule oxaloacetate. This releases the CoA and forms a 6-carbon molecule called citrate. Number 2. Citrate is converted into isocitrate by removing a water molecule and adding a new water molecule back. Number 3. Isocitrate is oxidized and releases a molecule of carbon dioxide. The remainder is a 5-carbon molecule called a ketoglutarate. As well, NAD+ is formed to NADH by reduction. Step 4. The 5-carbon molecule is oxidized, reducing NAD+ to NADH and releasing carbon dioxide. The 4-carbon molecule that remains picks up CoA to form succinyl-CoA. Step 5. The coenzyme A of succinyl-CoA is replaced by a phosphate group which is transferred to GDP, forming GTP as a product. Succinate is the 4-carbon molecule produced in this step. Step 6. Succinate is oxidized, forming another 4-carbon molecule called fumarate. Also, 2 hydrogen atoms and their electrons are transferred to FAD, producing FADH2. The enzyme that carries out this step is in the inner membrane of the mitochondria, so FADH2 can transfer its electrons directly into the electron transport chain. Step 7. Water is added to the molecule fumarate. This converts fumarate into a 4-carbon molecule called malate. And finally, step 8. Oxaloacetate, the starting 4-carbon molecule, is regenerated by the oxidation of malate. Another molecule of NAD+ is reduced to NADH. A single cycle nets out 2 molecules of carbon dioxide, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP or GTP. These numbers can be multiplied by 2 to find the per glucose yield, since these figures are for one turn and each glucose produces 2 acetyl-CoA. The electron transport chain can be described as a collection of membrane-embedded proteins and organic molecules organized into 4 parts, 1 through 4. As electrons travel through the chain, They go from a higher energy level to a lower energy level. Energy is released in these electron transfers, and several of the protein parts use the energy to pump protons from the matrix to the inner membrane, forming a proton gradient. All of the electrons that enter the transport chain come from NADH and FADH2 molecules produced in the previous stages of cellular respiration, which are glycolysis, pyruvic oxidation, and the Krebs cycle.